Good afternoon, viewers at home. My name is Olatunde Michael, the team leader of Omega Bio-Edutainment. Today, this is another uh, course under the educational series of Omega Bio-Edutainment. And today, we are going to take a course which is Microbial Evolution. Microbial Evolution. Okay, so today we are going to start with what is evolution. What is evolution? Just like I said before, it is believed that our heart is about 4.6 billion years old. Scientists believe that our heart is between 4.6 billion years, that is between 3.5 million years, billion years, that is BYA, or between 3.5 BYA and 4.6 billion years. And this is undoubtedly because, don't forget that I told you that evolution, what is evolution? Evolution actually means development with advancement. Development with advancement. Don't forget that evolution is one of the major characteristics of all organisms, including man. And we actually believe, scientifically, we actually believe that evolution is a powerful tool in speciation. Evolution is a tool for speciation. So what this means is that no species will ever exist without evolution, because if there is no evolution, there can never be life. So we believe that all organisms, according to Carl Woese, who believes so much that there are three domains of life. So the first domain is archaea, the second domain is prokarya, and the third domain is eukarya. Okay, what this means is that eukarya is the first to exist. Eukarya is the first organism to exist on the surface of this earth. Okay, this is the first, then followed by the second, and eukarya is the third. Now, in terms of multicellularity or the arrangement of cell of this, it is believed that this is going to be the most primitive. This is going to be the most primitive, followed by this and the most advanced, which consists of human, that is Homo sapiens, is part of eukarya, is the most advanced. So what this means is that microbial evolution and microorganisms can only be found majorly here. Let me say 80% of microorganisms will come from prokarya, whereas this one, let me say, we still have around 10% to be to consist of microbial world. So, and eukarya, we just have eukarya. This one is a single, is always a single cell or unicellular, just like a typical bacterial cell is unicellular. This one, because it is highly primitive, what this means is that even at unicellularity level, this one is not actually ripe to be called a unicellular organism, because this is the group that consists of prions. These are protein particles. They are just particles. They are not even living. don't forget that the first set of classification of organism actually believe that organism can be classified into acellular and cellular. That is, organism that comprises of a cell, and those ones that cannot actually be regarded as comprising of cell. So it is from this acellular that the group archaea actually evolved from. So because that is the group that consists of prions, and those ones that consist of virions. The other time I told you what are virions. These are virus. They are virus-like particles that are too primitive to cause any infection. But how did Carl Wilson actually come across this one? He used what is called SSU ribosomal RNA information. SSU ribosomal RNA. This S actually means simple subcellular unit of ribosomal RNA, because I told you that life actually started from DNA. Then DNA will undergo the process called transcription, and when this one is transcribed, they are going to have, so this is going to be double stranded, so it is going to be transcribed. It's going to undergo transcription into RNA, which is going to be single stranded, and from this RNA protein will be formed. This is a theory that we are actually familiar with. It is called the central dogma theory. So the question now is, after Carl Wilson's postulates that actually established all organisms to have arisen from one common ancestor, and that common ancestor, they use what is called LUCA. LUCA is the last universal common ancestor. That LUCA is what is here, which is the root. So just like I used to tell you that as a student of biology, you must believe in evolution. And don't forget the two typical theory of evolution, that is the theory of use and disuse, according to Jean Lamarck, and the second one, which is the law of Charles Darwin, that actually talk about survival of the fittest. So what that means is that I told you that initially E. coli, that is Escherichia coli, which is a microorganism, which is a bacteria, is common inhabitant of the human intestine. Okay, so because it is what causes gastrointestinal discomfort. So, but that is where the natural habitat of this microorganism. But nowadays, E. coli has actually migrated away from human guts, and you cannot find them in the environment. When they are found in that environment, that means the E. coli must have evolved and developed some certain characteristics or traits that will make them to be able to adapt well into their new environment. And that is where the law of Charles Darwin is actually coming into play. That is survival of the fittest. That is telling you all that whenever an organism, be it unicellular or multicellular organism, find themselves in a new environment, they must know how to be able to conquer that environment by developing some traits or some characteristics that will make them to be very, very suitable and adapted into that environment. And that is exactly what is here. So, and before this E. coli will migrate from habitat A into this their new habitat, which is habitat B, that means there must have been some changes in their genetic makeup. There must be some changes in their genetic makeup, and that is why I said evolution is a powerful tool for specification, for speciation. And what that means is that don't forget that the species of E. coli now, the species of E. coli now that actually migrated from habitat A to habitat B now, genetically they are going to be very, very different. That means it's going to take some new set of antibiotics to be able to actually treat any infection that is caused by this new organism. So that is what you should know. So what you should take home from here is that evolution is a powerful tool for specification, and that our world is between 3.5 billion years ago, years old, and 4.6 billion years ago. Now, the question is now the first organism. The first, when did we have said that the first, our heart is around this 3.5 billion years to 4.5 billion years ago old. So the next question is the first organism that existed on the surface of the earth. How did this organism actually look like? The first cell that first of all existed in the surface of the earth, was it like in form of unicellular organism? Was it in form of a cell or in form of a particle? Don't forget that according to a theory, operating theory of the origin of life, what he believed was that the first organism that existed was, because the surface of the earth then, we have there are 9 planets. There are 9 planets, and the earth is the third. And don't forget that it is still the third one. It is still the third. It's still the third where life was actually first reported. So by that time, Earth was not like this. It was very, very hot, because at that point in time, Opalin believed that our Earth was very close to the sun at that point in time, and because it was very close to the sun, it now made life on the surface of Earth to be very, very hot. And that was why the first life was said to be in form of a prebiotic soup. That is, it existed in form of a prebiotic soup. And I told you the meaning of pre. Pre is before or primitive. And when you say biotic, that is life. And why do they call it soup? You know, a soup is actually made of different ingredients, not just one. That time, that soup actually consisted of silica gel. It existed in form of silica gel, and that is that. Okay. So the first life, was in form of silica gel, and the time it existed was around 3.5 billion years ago. You need to actually know that. So immediately after the belief of prebiotic soup. So that prebiotic soup was like this. It was like this. And by the time it was like this, there was a kind of RNA that was coming to combine with the prebiotic soup, and that one actually led to what is called the RNA word. The first word was believed to be consisted of RNA word, and that was what actually happened. Okay, so when this now consisted, when this that one now consisted of RNA word, that is RNA now mixed with a prebiotic soup. Okay, now what now formed was protein, and that is why when you collect all microorganism or all organism, if you collect them till today, you inside microorganism you found RNA, you found DNA, and you found lipids. You found lipids, and these are part of what actually consisted of what this. So the RNA world then, because I said they believe that all organisms consisted of RNA. At that point in time, there was no clear definition of central dogma theory, whereby they believe that a DNA, which is a double-stranded, will undergo a process of transcription, and it is that transcription that will now form RNA, and from that RNA, you now have protein. Don't forget that protein is actually what made the world to be like. So that was what this was the belief initially about RNA world. They believed that RNA world existed, where all organism was made up of RNA, and this one was coming from prebiotic soup, which I said, okay, prebiotic soup, and the prebiotic soup actually consisted of lipids and silica gel. So by the time this now combined together, instead of it to form organism, so what it formed was the protein, and it is that protein, from that protein, that you see a lot of enzymes, and that one will, so it is all these protein that will catalyze metabolic reactions. It's going to be the one that will catalyze metabolic reaction. So immediately after, now let me now tell you about the three school of thoughts. The three school of thought. The first school of thought was the prebiotic soup, that all organisms consisted or was made up of soup in form of lipid. And at a point in time, the second school of thought was about the RNA world. So, and this was how RNA and prebiotic soup actually existed and combined together to form protein. And scientists now believe that this protein will now catalyze, because you know that all enzymes, they are protein in nature, but not all proteins that are enzymes. Okay, so this one now combined together to catalyze certain metabolic processes. So after prebiotic era, we have the era of RNA world. So after the era of RNA world, we now have microbial diversity. Microbial diversity. That is the era that actually believe that all microorganisms, they consist of different metabolic products that can make them to support life. Okay, so although this metabolic diversity era still believe that life has not actually existed as a single, that is, not as a single cell. So we need to know about that. Then again, immediately after I've told you about the prebiotic soup era, the RNA world era, and the metabolic diversity era, the next one now is the era of endosymbiosis. The era of endosymbiosis. So don't forget that endo actually means inside. Endo means inside. Endosymbiosis. Okay, I told you that in biology, there are certain things that you will even know when you try and understand from the simplest rule of okay, for example, now in endosymbiosis, endo means inside or internal. Syn is coming from symbiotic association, that is similar. Similar. Syn. So then bio means life. Okay, and cis is actually coming from splitting. Okay, so that means during that time of endosymbiosis, we believe that some certain cells they existed as single cell, unicellular. Some of them were small, some of them existed as a very big cell, and at the end of the day, very big cell. So as a result of, don't forget that each cell has its own metabolic products. Each cell has its own level of existing independently. So for example, now let's say this one is has the function of carrying out protein synthesis. If this one is responsible for protein synthesis, and this one is just a vacuole, it's just a vacuole, okay, just empty. Then there are some that they have photosynthetic functions. So, and there are other cells that they have their own function in carrying out what is called cellular respiration. Cellular respiration. So if all these cells now existed, 1, 2, 3, 4, each of them have their own function. This one, its function is that it is very big, that it can actually house all these, the other three. So this one has a function in terms of protein synthesis. Now, if you now trace it back to our modern day cell, or what we call organelle, you will believe that the organelle that is responsible for protein synthesis in the modern cell today is ribosomes. Ribosome, or your mind will be going to endoplasmic reticulum. So at that point in time, this one that was said to be big for nothing, at that point in time, actually swallow all the other small ones. And this one that you can say that they are big for nothing is what is now our modern day plasma membrane that serves as the organelle for delimiting, organisms. I mean, for delimiting certain activities of the cell. And those ones that carries out photosynthetic function, that one will photosynthetic function will go to definitely will go to plant cell. Whereas its contemporary, which is actually responsible for cellular respiration, will go into animal cell. So what the theory of endosymbiosis actually believed was that all the cells existed because they could carry out different types of metabolic processes. And what are these metabolic processes? Cellular respiration is part of them, of course, and that's when you'll be thinking, I'll be talking of Krebs cycle, glycolysis, and so on. Then photosynthetic, that is when you'll be talking of Calvin cycle and some others. Then protein synthesis, which has to do with protein synthesis as we have it today, which is a process of translation. Okay, now this bigger one, as a result of the theory of endosymbiosis, now swallow all this other one, and by the time it swallow it, because it is a symbiotic association, not a parasitic association, the swallowing did not actually affect each of them to carry out their independent function. So, and that's why our modern cell today consists of organelles. Organelles actually means small organs that are very independent and they can go on with their own metabolic processes. So, and that is the theory of endosymbiosis, and it is on this theory that we now have the photosynthetic function was carried out by chloroplasts, and that is what is known as a chloroplasty today, and that of cellular respiration is what is actually known as a mitochondria today. Okay, so that is, so I talk about prebiotic soup era, RNA world, the era of metabolic diversity era, and the modern day era. I mean, sorry, the endosymbiotic era, and now it is now we now have the modern day microorganism. And whenever we talk of microorganism, what you should actually talk of, or the image as it come into your mind, should be a typical bacterial cell, that carries out a different function. And don't forget that most of what prokaryotic life, or most of what prokaryotic cell does is still what eukaryotic cell also do as well. Okay, so the next thing is phylogeny. So how did you come about phylogeny? Whenever you come across phylogeny, so another word for phylogeny is okay, whenever you come across phyllo is actually talking about, okay, and geny from genetics. So that is now what you now call as the possible to draw to draw phylogenetic tree, that is for you to be able to draw a tree that will represent the origin of a human. So that is a very important aspect of this course. You should be able to know how to draw phylogenetic tree. It's very important. For example, now if I draw a phylogenetic tree like this, okay, so if this one is A, if this one is B, if this is C, if this is D, if this one is E, if this one is F, so it has, you need to know how to interpret this tree. A is regarded as the ancestor, which is the origin of all of them. There are two basic types of phylogenetic tree. I need to tell you that. There are two basic types of phylogenetic tree. The first one is called rooted, and the second one is called unrooted. The first one is called rooted, and the second one is called unrooted. The rooted one is the example of rooted phylogenetic tree is what I have just drawn on the board. That all of them, they have one origin, which is their common ancestor here, which is this. The unrooted one, I'm going to give you example of that as well as we move on. Now, okay, I said A, B, C, D, E, F. If you want to interpret this, I told you that A is the common ancestor for organism A, B to F. Whereas at this point before the branch are called the node. You call them the node. So node B, node C, and D, just like that. So this one is also node. This one is just also node as well. So whenever you are talking of, that's why they call it a tree, because you see the structure is just like a tree. You imagine this as the trunk. You imagine this between A and B, you imagine it as a trunk. All these one are branches, so you need to know that. Now let me now draw, this is a typical rooted phylogenetic tree diagram. Let me try and draw the unrooted one, so that you'll be able to understand how to interpret them. Okay, Okay. Now, let us say this place is called the roots. This place is called the roots, and from here to this place, just like I told you, is called the node. Okay. Then all these points, we call them the node, just like I told you here. So from the line, this line is called the branch. We call it the branch, and here, okay, so this place is called the taxon. Taxon is the Latin form of the group. So another word, the English word for taxon is called the group. Okay. So this is a species. Is a species, and that is a species F. Don't forget, there's no singular and plural of a species. Some of you will say because it's just one, you will pronounce it as species. No, species is species. It's not like in English. Thereby you have to pluralize some. So this place now, so this is A, this is species A, this is species B, this one is C, this one is D, this one is E, and this one is F. So I've told you that these points are called the nodes. So just like you have them here. So now you can see that from here to here is another node, and from this another node you now have another one. This is called the external node. External node. Okay. Then you will see that D and E, D, E, C, you can see that they are from the same branch, like that. C, D, E, they are from the same branch. So C, D, E are called clade. Call them clade because they come from the same what? They come from they are called clade. Then this one is called the SB. They are called the external node. So it is now right to call this one, all these one are called species. So what do we mean by species? If we say species in biology, species actually means set of organisms, set of organisms with similar characteristics, set of organisms with similar characteristics that can freely interbreed. That is what is called a species. So according to Charles Darwin, he now believe that sometimes you have some set of organisms that they belong to the same species, but at a point in time, as a result of divergence between two of them, they will get to a point interbreed freely. So that is where when you now call them as a subspecies, or what is known as strain. So these are part of what you should know in this course. And finally, to conclude this aspect, why I actually give you all this background is just that evolution is the roots, or is a very powerful tool for speciation, and that without any change, evolution cannot actually take place. And you should believe that whenever there is evolution, that means there is a surety that there must have been some certain genetic rearrangement, genetic change. And I told you that there are two basic types. There are two basic types of the mechanism, different organism coming. So you can have them as in form of horizontal gene transfer, and it can be in form of vertical gene transfer. We are going to see this in subsequent lectures we are going to have on